Good day, this is Professor Will, CBMD PhD, case Friday, July 19, 2019. It is 2:33 and 2 seconds Eastern Standard Time. And this is a continuation of my scientific research in pathophysiology. So let's get started here. Microorganisms are prime movers in all ecosystems. They mobilize some elements by converting them from non-volatile to volatile forms, permitting large-scale mass transport of these elements among different geographic locations. They capture carbon dioxide and other one-carbon compounds through several known fixation pathways, accounting for approximately 50% of global primary productivity. Microorganisms participate and frequently dominate the interconversions of chemical compounds, producing and regulating the great variety of materials necessary to the biosphere. Effectively, all decomposition processes, including spoilage of foods, are dominated by the microbiota. Microorganisms are found in all environments in which liquid water and energy source can be found. These include such extreme environments as polar sea ice and fluid-filled channels and pockets, hydrothermal vents, hot and cold deserts, brines approaching salt saturation, extremely acidic and alkaline waters, and deep 1 kilometer subsurface solid rock formations. It can be accurately stated that the Earth is a microbial world. Microorganisms also participate in a plethora of symbiotic, literally living together, interactions with each other and with higher organisms. Some are common commensals, using the products of their host without causing any discernible harm. Some form mutualistic interactions in which they and their host both actively benefit from the symbiosis. These mutualisms range from production of essential vitamins and facilitation of digestive processes by the microflora found in the digestive tract of animals to the elegant and species or strain-specific nitrogen-fixing interactions that occur between many plant species and bacteria. Finally, microorganisms are infamous for their parasitic interactions with higher organisms. These range from subtle, such as influence of Wolbachia infections on sex determination in arthropods, to catastrophic. The impacts of pathogenic microorganisms on human populations have on occasion been devastating, and the plagues that ravaged Europe and Asia throughout the Middle Ages are no more notable in this regard than the AIDS pandemic of the present. The impacts of parasites on the structuring of higher organisms communities can obviously be very significant, and as Dutch elm disease demonstrates, even a single pathogen can drive a host species to local extinction. The exploitation of microorganisms having useful properties is essential to many human enterprises and has fostered substantial bioprospecting efforts. Numerous microbial products ranging from antibiotics to foods are important commodities, but perhaps the most significant impact of beneficial microorganisms lies in agriculture. Many microorganisms that grow in mutualistic interactions with plants have been used to increase crop yields and to control pests. Nitrogen-fixing mutualisms between plants and bacteria form the basis for crop rotation and can facilitate reclamation of overexploited or marginal lands. The mycorrhizal fungi, which grow in intimate association with plant roots, enhance the ability of plants to take up mineral nutrients and water. This type of symbiosis permits plant growth in many marginal environments, and most plants participate in it. Numerous bacteria, non-mycorrhizal fungi, colonize the surfaces of plants and can successfully outcompete potentially pathogenic organisms, preventing infections. In addition, the highly specific insecticidal toxins produced by Bacillus thuringiensis have been harnessed in insect pest control strategies and offer the major advantage of not harming beneficial insect species. Agriculture is and always has been highly dependent on microorganisms. The vast array of microbial activities and their importance to the biosphere and human economies provide strong rationales for examining their diversity. Loss of biodiversity is an ongoing global crisis, and loss of microbiota indigenous to human-impacted environments are associated with higher organisms facing extinction is a real concern. As a consequence of the growing recognition of the importance of species loss, achieving a meaningful assessment of microbial diversity has been the topic of several major workshops and meetings. This is a period of very active inquiry into microbial diversity. Past studies of microbial diversity were highly dependent on our ability to isolate these organisms into pure, i.e. single species, laboratory cultures for characterization. However, in most environments, only 1 to 5% of microscopically viable bacteria and archaea can be successfully cultivated, and obtaining pure cultures of many alga, protozoa, and fungi is problematic. In addition, detailed morphological, physiological, and behavioral characterization of any microorganism can be extremely time-consuming. As a consequence, many pure culture isolates have only been partially characterized. Recent applications of molecular biological tools, chiefly polymerase, chain reaction, PCR, DNA hybridization, DNA cloning, and sequencing, and efficient analysis of DNA sequences. I have provided a means to catalog microorganisms without the necessity for isolation and offer useful methods for streamlining the characterization of pure cultures. Commonly used strategies for accessing microbial diversity employ PCR to amplify specific bacterial genes extracted from samples from the environment of interest. Nucleotide sequence of genes encoding a small subunit ribosomal RNA, 16S rRNA for bacteria and archaea, and 18S rRNA for the eukarya. When properly aligned, provides substantial information on the relatedness of unknown organisms to known species and support the construction of phylogenetic trees for microorganisms. Other genes of interest for characterizing microbial diversity encode key enzymes involved in microbially medicated processes. The amplified gene sequence can be resolved using various gel electrophoresis methods to provide a fingerprint of the microbial community present in the sample, or they can be cloned into an appropriate vector for propagation and nucleotide sequence analysis. The sequences of these genes are then available for phylogenetic analysis, and species-specific domains within them can be used in DNA hybridization studies to quantify organisms of interest in environmental samples. Similar phylogenetic analysis of uncharacterized pure cultures allow the relatedness of these organisms to known species to be efficiently determined. Knowledge of the type of organism studied greatly facilitates selection of appropriate testing criteria for its complete identification. These technologies, although not perfect, have opened the door to the vast unknown world of microorganisms, clarifying the relationships of microorganism groups to each other and to higher organisms and facilitating the catalog of species that have thus far resisted isolation and cultivation efforts. It is also clear from recent ongoing studies that many new taxonomic lineages of microorganisms await discovery. The bacteria constitute an extremely diverse domain of prokaryotic organisms. They have a very simple cellular architecture lacking the membrane-bound organelles characteristic of eukaryotic cells. They reproduce asexually, typically by simple fission, and are genetically haploid. Bacteria acquire soluble nutrients by transport across the plasma membrane, but are unable to engulf colloids or particles through any endocytosis mechanisms. Virtually all known bacteria taxa have cell walls composed of an amino sugar polymer called peptidoglycan, and this material is unique to this domain. Numerous distinctions can also be found between the bacteria and other types of organisms and the details in their information processing machinery. Important example: DNA replication, RNA synthesis, and protein synthesis systems, chromosome structure, flagellar structure, and many physiological features. Although unified by many features of cell structure and function and clearly forming a monophyletic domain, the bacteria encompasses enormous physiological diversity and can be found in almost every conceivable ecological niche. These organisms are found in extreme environments ranging from Antarctica sea ice to superheated marine hydrothermal vents, from extremely dilute aqueous systems to solutions approaching salt saturation, from highly acidic waters approaching a pH value of 0 to alkaline waters with pHs in excess of 12, and from the near vacuum of the upper atmosphere to the crushing pressures of the Challenger Deep. Bacteria can even be found in the highly radioactive environment of nuclear power plant reactor chambers. Although a few very hot environments appear to be dominated by archaea, the bacteria are ubiquitous inhabitants of all other environments and are important, and sometimes the only participants in numerous ecological processes. Bacteria dominate mineral cycling and degradative processes in most environments, are important primary producers. The bacteria dominate the biosphere like no other type of organism on Earth. Good day, this is Professor Will, CBMD PhD. Today is Friday, July 19, 2019. It is 4:34:40 Eastern Standard Time. And this concludes my reading on pathophysiology and microbial diversity. Hope you enjoyed listening. Have a great day.